So it's time to get started on Chapter 4, and in this chapter we're going to be looking at the characteristics of prokaryotic and eukaryotic cells. So there are two large, and they're considered non-taxonomic categories of cells. So by taxonomic we mean talking about evolutionary relationships. Now we're looking at just classifications based on the way that the cells appear, the characteristics that they have. And so one of these categories is prokaryotic cells. These are the bacteria and the archaebacteria. So they're small, they're single-celled, they don't have a nucleus, and they don't have the other membrane-enclosed organelles that we find in eukaryotic cells. So again, domains bacteria and domain archaea together are the prokaryotic groups of cells. Eukaryotic cells are larger and tend to be much more complex. They can be single-celled. Many of them are also multicellular. They have a nucleus, so there is a region of the cell that's bounded by membranes that stores the DNA. And they also have other membranous organelles within the cell. And the eukaryotic cells all belong to domain eukarya, which includes the plants, animals, the funguses, and all of the protist kingdoms. And there are multiple kingdoms. So these are all kingdom level terms. So there's kingdom Plantae, kingdom Animalia, kingdom Fungi, and then there's a number of kingdoms that together are considered protists. And then last of all, there are non-cellular, non-living structures that are the viruses. And so we'll talk very briefly in this chapter about viruses. Viruses are acellular and they are not living, and depending on the organisms, some specialize on prokaryotes while others specialize on eukaryotes. And here we have a cartoon showing the characteristics associated with a prokaryotic cell. The other cartoon is showing the characteristics associated with eukaryotic cells. So you can see eukaryotic cells much, much more complex, way more structures in them. So, as you've heard me say, there are three domains. These are three major groupings, evolutionary groupings of organisms. And the domains are called domain eukarya. Those are the eukaryotic cells, the things that we think of when we think about living organisms most of the time. So within the within the domain eukarya, we see a number of different kingdoms. There's kingdom Animalia, kingdom Plantae, kingdom fungi, and then we have these organisms that are the protist kingdoms. And we're still working a lot of this out. With the advent of the ability to look at and sequence DNA, we're seeing that what we used to think of as a single kingdom is actually more than one kingdom, and there's a lot of work that's being done on that right now. Domain archaea, these are also known as the archaebacteria, and anytime you see an A and an E together, recall that you pronounce E with that, so that's why it's archaebacteria and domain archaea. So the domain archaea, they are prokaryotic organisms, and they are frequently found in extreme environments. So places like deep sea vents at the bottom of the ocean, or in highly sulfurous waters, which would be very, not conducive to the growth of many organisms, or in places where there are really extreme temperatures. So in some of the really, really hot springs, or in glacier ice, or in ice that you can drill down to, so really extreme places tend to be the locations where we see the archaeans, but that is a generality. And then last of all, there's domain bacteria. These guys are also prokaryotic, and they include most of what we think of as the free-living and pathogenic bacteria. So two domains, and again, these are the protist domains. So bacteria, archaea, the protist, I'm sorry, no, I said protist, I meant prokaryote. the prokaryote domains. And here in purple, we see a bunch of the groups of bacteria, and here in red, we see some of the groups that we associate with the archaeans. Okay, so we'll start by looking at the characteristics of prokaryotic cells. Prokaryotes are among the smallest of all the living organisms, with sizes that range between a half all the way up to 2 millimeters. I'm sorry, 2 micrometers. So this weird, like M-looking structure, it looks like this. It's like a cursive M with a long tail here. Oh, now I've really screwed it up. Let's try that again. There we go. That means micro, so that's 10 to the minus sixth. So we're talking about a half of 1/1,000,000th of a meter to all the way up to 2/1,000,000ths of a meter in diameter. And because they are so small, they have what is considered a high surface area to volume relationship. So when you think about the whole volume of an archaean or a prokaryote, let's say a spherical bacterium like these guys here, the surface area, that's the cell membrane that surrounds the entire cell. So that would be its surface area. The volume would be all of the fluid plus the organelles and other parts that we find inside of the cell. So when we say that an organism has a high surface area to volume ratio, that means that there's, it's only a short distance to get from the furthest part away where the, where living parts of the cell are found to the membrane where we exchange gases and exchange nutrients and wastes. And so because prokaryotic cells have such small bodies, and that means that they then have lots of surface area for every unit volume of them, then that nutrients and wastes can move very quickly just by the physical properties of the cell. So using properties like diffusion, which we'll get to later into this chapter, is sufficient to move materials throughout the cells because of this high surface area to volume ratio that they have. Prokaryotic cells come in a variety of shapes, and here are 6 of them. If it's a spherical, sort of a ball-shaped cell, then that is a coccus, or we would say cocci if we were talking about them in plural. So the bold word is the singular form, the non-bolded form is the plural word. A bacillus is a rod-shaped, so these are spherical here. These are rod-shaped. And a coccobacillus is sort of in between a coccus and a bacillus. So it's a stubby rod or a slightly elongated coccus. And so they get that classification kind of in between the two of coccobacilli. A spirillum is one that shows this rigid wavy shape. So it's not a corkscrew. Instead, it's just this a waviness that we see here in this image. A vibrio is a little comma-shaped or slightly curved rod-shaped prokaryotic cell. And then last of all are the spirochetes. Spirochetes are helical, so they are like a corkscrew is the shape that they have versus the spirillum, which is just a wavy shape. Okay, so 6 different shapes. These aren't the only shapes. There are more, but these are the most common shapes that we see amongst the prokaryotic cells. Most organisms are what are considered monomorphic. Mono meaning single or one. Morphic is about the shape. So morphology is the shape of something. So a monomorphic cell or colony or species is one in which there's only one shape in the culture that we're growing of these prokaryotic cells. Pleomorphic, on the other hand, means that there are multiple shapes found within a colony, or multiple characteristics to the shape of the cells. And we can see pleomorphic bacteria even among cultures that started with a single cell. And it tends to be, so the general rule is that older cultures are more likely to be pleomorphic. And frequently they have smaller cells as well. And so as a colony or a culture begins to age and they start to use up all the nutrients that are available to them in their culture medium, then they tend to become more pleomorphic and smaller in general. And here is an example of a bacterium called Mycobacterium. And they're using with this an acid-fast stain. And note that some of the organisms in this pleomorphic culture stain as acid-fast rods. They're the ones that look kind of pinkish, reddish there toward the center. And then some of them stain as non-acid-fast cocci. So there's an example of pleomorphism amongst the Mycobacteria. So not only is it the shapes that you want to keep track of, but also the arrangements. So when a prokaryotic cell divides, it divides via a process called binary fission. And in binary fission, the chromosome duplicates. So we see a second chromosome form. So if this is the cell, then, and here's one of the chromosomes, and it's attached here at the membrane, then another chromosome forms. It's also attached to the membrane, and over time, as the cell grows, the two chromosomes are going to separate from each other because we're adding more cell membrane here in between where they are attached to each other. And so finally, the cell will pinch into two, and we get two sort of smaller rod-shaped bacteria. So when they pinch into two, they may or may not divide, or separate from each other. So they divide, but they may or may not separate. So amongst the cocci, if they don't separate, and the cells are found in pairs, then they're referred to as diplococci. So di meaning two. So here's some diplococci. Streptococci they keep dividing and remain attached to each other until they form long chains of cells or strings of cells. So those are the streptococci, so like Streptococcus aureus, for instance. Sometimes they will divide until there are four of them all attached to each other, and they're arranged sort of like a square. Those are referred to as tetrads. So here are some tetrads. A tetrad, tetras or tetra, that means four. Sarcini, and that's how you would say that if it's plural, sarcina or a sarcini. Sarcini are when the cells occur in groups of 8, and so they form not just a tetrad, but with two tetrads sitting on top of each other, they form a cube. So a total of 8 cells. And then if it's Staphylococcus, then we see grape-like clusters. So a whole bunch of them kind of all wadded together like grapes that you might buy at the grocery store. So no particular arrangement to them if they are Staphylococci. The bacilli can, as they undergo binary fusion, if they remain attached, then they can form these long chains. They look like train cars back to back to back, and those are the streptobacilli. And they can also connect side to side. And I don't know that there's a particular name for that one. I couldn't find anything about that. Among the spirilla and the spirochetes, they tend to separate completely. So we don't see this kind of hanging together that happens with them.